This is a second lecture on endocrine glands. In this lecture, I will be talking about hypothalamus, pituitary, and thyroid glands. Hypothalamus is a major link between nervous and endocrine system. And some of the endocrine glands are controlled by hypothalamus by the secretions from the neurosecretory cells. This is an enlarged view of hypothalamus and pituitary glands. This is hypothalamus, which has got neurosecretory cells which secrete the hormones or chemical factors which control pituitary. And this is pituitary. It has got two parts, posterior pituitary and anterior pituitary. And this pituitary gland is connected to the hypothalamus by infundibulum, through which the connection is established and the secretions from the hypothalamus reach pituitary glands. So anterior pituitary is also known as adenohypophysis and posterior pituitary is also known as neurohypophysis. Anterior pituitary release hormones by a stimulation or inhibition messages from hypothalamus. Also regulated by negative feedback. Secretions of anterior pituitary is also controlled by the presence of the hormones in the blood. If the hormone is less in quantity, less in concentration, this anterior pituitary will secrete more hormones and release into the blood. If the blood is having enough concentration of these hormones, the secretion will be inhibited. In hypothalamus, hormones are made by neurosecretory cells, which will be transported to the pituitary by hypophyseal portal system. Anterior pituitary hormones that act on other endocrine systems. Some of the hormones from the anterior pituitary acts on other endocrine systems. They are called tropic hormones. So they control the functioning of other endocrine glands. The hormones from the anterior pituitary glands. Human growth hormone, as the name implies, it supports growth of human beings or growth of an individual and is also called somatostatin. It stimulates secretion of insulin-like growth factors that promote growth and protein synthesis. You know that for the growth of human body, more cells need to be made, and for this, protein is important. And most of the metabolic reactions is catalyzed by enzymes, and enzymes are proteins. As a result, growth hormone induces secretion of insulin-like growth factor (IGFs), which promote growth and protein synthesis. Then next hormone is thyroid stimulating hormone or thyrotropin. From the name itself, we can say that it stimulates thyroid glands to secrete the hormones of the thyroid gland. It promotes the synthesis and secretion of thyroid hormones. Then follicle stimulating hormone or FSH. It has effect on ovaries as well as testes. In ovaries, it initiates the development of oocytes. In testes, it stimulates testosterone production. As you know, testosterone induces the secondary sexual characters in man. Luteinizing hormone, this also has effect both in man and women. In ovaries, it stimulates ovulation and stimulates the production of estradiol and helps to control menstrual cycle. After fertilization, LH stimulates the corpus luteum, which produces progesterone to sustain the pregnancy. In men, luteinizing hormone stimulates the production of testosterone from Leydig cells in the testes. Testosterone, in turn, stimulates the sperm production and helps to accentuate male characteristics like deep voice or growth of facial hair. Prolactin promotes the milk secretion by mammary glands in mothers. Adrenocorticotropic hormone or corticotropin. ACTH stimulates glucocorticoid secretion by adrenal cortex. Melanocyte stimulating hormone or MSH, its exact role is unknown in human beings. So this is a diagram showing the control and secretion of hormones from the anterior pituitary. The start is hypoglycemia. It stimulates the release of growth hormone releasing hormone GHRH from hypothalamus. So this growth hormone releasing hormone stimulates the release of growth hormone by anterior pituitary. Increased growth hormone and insulin-like growth factors speeds up glycogen breakdown into glucose so that more glucose is available for metabolism and for the energy release. Increased growth hormone and insulin-like growth factors speed up glycogen breakdown into glucose, which enters the blood more rapidly. So this provides the fuel source for making enough ATP for the increased metabolic reactions for growth. Blood glucose level rises to normal. If this increased release of glucose is causing hyperglycemia, it will inhibit release of GHRH, growth hormone releasing hormone. Hyperglycemia induces the release of growth hormone inhibiting hormone. When hyperglycemia is caused, it suppresses the release of growth hormone releasing hormone. At the same time, it induces the release of growth hormone inhibiting hormone by the hypothalamus. Growth hormone inhibiting hormone inhibits the release of growth hormone by the somatotrophs. Low level of growth hormone and insulin-like growth factors decrease the rate of glycogen breakdown. Less glucose will enter the blood. Blood glucose levels falls to normal. If the decreased glucose release leads to hyperglycemia, it will induce the release of GHRH. The synthesis and release of growth hormone releasing hormone and growth hormone inhibiting hormone is controlled by hyperglycemia or hypoglycemia. During hypoglycemia, growth hormone releasing hormone will be released, which causes the production of or which stimulates the production of growth hormone in the anterior pituitary. This in turn will increase breakdown of glycogen and glucose will be released into blood and hypoglycemia will be controlled or resolved. During hyperglycemia, growth hormone releasing hormone production will be suppressed and instead another hormone will be produced by the hypothalamus, which is growth hormone inhibiting hormone. This will be released into the blood and this reduces the glycogen breakdown into glucose. As a result, glucose supplied to the blood will be decreased and hyperglycemia will be controlled. If this production of growth hormone inhibiting hormone and resultant decrease in the growth hormone results into hypoglycemia, again it will go back to the increase in the growth hormone releasing hormone. Posterior pituitary does not synthesize hormone, but it stores and releases hormones produced by hypothalamus. The hormone synthesized and released by the hypothalamus is transported along hypothalamo-hypophyseal tract. Oxytocin and antidiuretic hormone or vasopressin reach the posterior pituitary from hypothalamus. Oxytocin during and after delivery of baby affects uterus and breast. So oxytocin enhances the smooth muscle contraction in the wall of the uterus, which facilitates parturition. Oxytocin also stimulates milk ejection from mammary glands. Antidiuretic hormone, as the name implies, it is against diuresis. It decreases the quantity of urine produced by controlling the water content in the urine. The decrease in urine production is by causing the kidneys to reabsorb more water to the blood. It also decreases water loss through sweating. Constriction of arterioles is another effect of ADH. This will increase blood pressure. How is the synthesis of ADH controlled? ADH synthesis and release is controlled by osmolarity of the blood. High blood osmotic pressure stimulates hypothalamic osmoreceptors. Osmoreceptors are present in the hypothalamus. Increased osmotic pressure of the blood stimulates these osmoreceptors. As a result, activation of neurosecretory cells occurs, which induces synthesis of ADH and ADH will be released. ADH is released from the axon terminals into the bloodstream. This reaches the kidney. As a result, water reabsorption in kidney increases. and concentrated urine will be produced. ADH also has effect on sweat glands. It decreases production of sweat and hence decrease water loss. So water is conserved. ADH reaches the arterioles and causes their constriction, which increases blood pressure. The osmolarity of the blood is normally increased by decreased volume of the blood or the decreased percentage of water in the blood. When the volume of blood decreases, it automatically decreases blood pressure. That is not good for the body. So to increase the blood pressure, the ADH constricts the arterial wall. As a result, arterioles become smaller and it increases blood pressure. So the blood pressure will be maintained. Low osmotic blood pressure inhibits hypothalamic osmoreceptors. As a result, ADH production will be decreased. This causes just opposite effect. ADH is less, water reabsorption returns to normal. Low osmotic pressure is normally due to increased water reabsorption in the blood. This can also cause increase in blood pressure. So water needs to be removed. In the absence of ADH, water reabsorption at the kidney will be decreased. So a dilute urine will be produced and excess water will be excreted through urine. And blood pressure will come back to normal. Thyroid gland is located below the pharynx. It has two lobes and these two lobes are connected by isthmus. Thyroid follicles inside the thyroid gland produce thyroid hormones. The major thyroid hormones are T4 and T3. Thyroxine or tetraiodothyronine. It is also known as T4. Tetraiodothyronine has 4 iodine atoms in one molecule of the hormone. Triiodothyronine T3 it has 3 iodine atoms in one molecule. Both these hormones increases basal metabolic rate. It stimulates protein synthesis. These hormones increase the use of glucose and fatty acids for ATP production. So the metabolism will be increasing. Parafollicular cells or C cells of thyroid gland produce calcitonin. It lowers the concentration of calcium ions in blood by inhibiting bone resorption. So less calcium ions will be released on the bones. How is the thyroid hormone controlled? Thyrotropin releasing hormone from the hypothalamus control thyroid hormone secretion. And thyroid stimulating hormone from anterior pituitary also controls the secretion from thyroid glands. In addition to these two hormonal control, increase in ATP demand also increases secretion of thyroid hormones. In the previous slide, we have seen that thyroid hormone increase basal metabolic rate, which can also release increased amount of ATP. During increased ATP demand, more thyroid hormone need to be released. As a result, increase in ATP demand also increases secretion of thyroid hormone. This is a schematic representation of the control of thyroid hormone release. Low T3 and T4 levels in blood or low metabolic rate or increased demand of ATP stimulate release of thyrotropin releasing hormone from the hypothalamus. So these conditions, low T3, T4 and low metabolic rate or increased ATP demand, increase the release of or stimulate the release of thyrotropin releasing hormone from the hypothalamus. TRH is taken to the anterior pituitary by the hypothalamic hypophyseal portal system. By the blood vessels, it reaches the anterior pituitary. And thyroid stimulating hormone synthesis will be stimulated and which will be released into the blood. This stimulates thyroid follicles to synthesize T3 and T4. This may increase the level of T3 and T4 in the blood, which will cause a negative feedback process. As a result, thyroid stimulating hormone production will be decreased. Production of thyrotropin releasing hormone from the hypothalamus is also decreased. TRH from hypothalamus is a tropic hormone. As I said before, it indirectly affects the cells by affecting the release of other endocrine glands. Here TRH induce the production of TSH by anterior pituitary. This TSH in turn increase the production of thyroid hormone. This thyroid hormone increase the basal metabolic rate in cells. So it's an indirect stimulation of cellular activities. So this TRH is a tropic hormone. The thyroid gland, anterior pituitary gland and hypothalamus. This system comprises a self-regulatory circuit which is known as hypothalamic pituitary thyroid axis. This is a self-control system. Calcium signaling in thyroid hormone production and release. Calcium signaling in TH production and release involves phospholipase C. Calcium signaling is a process whereby the extremely low cytoplasmic Ca2 plus concentration increases. This increase is by the release of Ca2 plus ions from the storage sites in the cell cytoplasm, like endoplasmic reticulum, microsomes, etc. It is done in a deliberate and specific manner to trigger downstream cellular events. Actually, every tissue and every cell types in human body utilizes some form of Ca2 plus signaling to function or survive. So Ca2 plus signaling is one of the most important controlling mechanisms in our cells. There are many mechanisms which can generate Ca2 plus signals. The most widely encountered is a mechanism involving a lipid metabolizing enzyme, phospholipase C. This is the only enzyme that can directly produce or moderate three distinct signals. These three distinct signals involve inositol 1,4,5 triphosphate or IP3, diacylglycerol DAG and phosphatidyl inositol 4,5 bisphosphate PIP2. Out of this phospholipase C signaling, initiating reaction is the breakdown of the lipid phosphatidyl inositol 4,5 phosphate PIP2 producing inositol 1,4,5 triphosphate. PIP2 is converted to IP3. This IP3 functions as a Ca2 plus signaling second messenger. All three phospholipase C derived signals regulate ion channels. So their effect or influence is made by regulating ion channels, mainly Ca2 plus channels. This conversion or these signals induce the release of Ca2 plus from intracellular storage, that is endoplasmic reticulum, microsomal fractions, etc. The release of Ca2 plus endoplasmic reticulum deplete endoplasmic reticulum Ca2 plus pool. This will activate Ca2 plus influx from extracellular fluid through plasma membrane. So this is how TSH release is controlled by phospholipase. The thyrotropin releasing hormone acts via phosphatidyl inositol 4-phosphate, that is PIP, protein kinase C and calcium signaling, that is shown in this diagram. Thyroid stimulating hormone or thyrotropin is secreted by the cells in the anterior pituitary called thyrotropes. This hormone binds to its receptors on epithelial cells in the thyroid gland. So the receptors are on these epithelial cells. This hormone binds to the receptors on the basolateral side of the epithelial cells and stimulate their gland to synthesize and release thyroid hormones. TSH, thyroid stimulating hormone, is a glycoprotein hormone. The most important controller of thyroid stimulating hormone secretion is thyroid releasing hormone. As already said, the thyroid releasing hormone is secreted by these neurons in the hypothalamus into hypothalamic hypophyseal portal blood. The receptors for thyrotropin releasing hormone are found on the thyrotropes in the anterior pituitary and stimulate secretion of TSH. One interesting aspect of thyroid releasing hormone is that the polypeptide has only 3 amino acids in it. Its base sequence is glutamic acid, histidine, proline. So only 3 amino acids are there in this hormone. Thyroid stimulating hormone TSH mediates its effects through its receptors, thyrotropin receptor or TSHR.
located on the basolateral surface of thyroid follicular cells, on the basal surface as well as on the lateral surface of these follicular cells. TSHR, that is receptor, is an integral membrane protein and is a member of G protein coupled receptors, G protein or guanine nucleotide binding protein. TSHR interacts with the G protein to initiate signaling. Thyroid stimulating hormone come and bind with TRH or TSHR receptors. This receptor is coupled with the G protein, that is guanine nucleotide binding proteins. This binding initiates the initial signaling. Binding of TSH with TRH receptors on thyroid epithelial cells stimulate production of iodine transporter, thyroglobulin and thyroid peroxidase proteins. So when this hormone is bound to the receptor, it induces or stimulates the production of iodine transporter, thyroglobulin and thyroid peroxidase proteins, which are essential for the synthesis and secretion of thyroid hormones. TSH binding to the receptor activate both the CIMB pathway as well as phosphatidylinositol or calcium second messenger signaling cascade. So this binding of TSH opens up or activates two pathways, one where the cyclic AMB is produced and the other one phosphatidylinositol or calcium second messenger system is activated. This second messenger system is a cascade of downstream processes finally leading to the production of thyroid stimulating hormone. When this phospholipase C is activated, it breaks down phosphatidylinositol 4,5-P, that is PIP. As a result, two-way the process will activate protein kinase C. One is the attachment with 1,2-diacylglycerol. The second is the formation of inositol 1,4,5-triphosphate, that is IP3, which is a second messenger. This causes the release of Ca2 plus ions from their storehouses in the cell like endoplasmic reticulum, microsomal particles, etc. And this will activate protein kinase C, which in turn will result in the transcription of the DNA, the gene for TSH hormone, and mRNA is produced. mRNA moves out of the nucleus and binds to the ribosome, and the TSH protein is produced, which is stored in vesicles. The vesicles move to the cell surface and break open and release the TSH into blood. This binding results into two types of signaling. One is calcium signaling, and calcium signaling involves phospholipase C. Calcium signaling is a process whereby the extremely low cytoplasmic Ca2 plus concentration increases. This is happening in a deliberate and specific manner and trigger downstream cellular events. Virtually every tissue and even every cell type in the human body utilizes some form of Ca2 plus signaling to function or survive. There are many mechanisms which can generate Ca2 plus signals. The most widely encountered is a mechanism involving a lipid metabolizing enzyme, phospholipase C. Phospholipase C is the only enzyme that can directly produce or modulate three distinct signals. That is inositol 1,4,5-triphosphate IP3, diacylglycerol and phosphatidylinositol 4,5-bisphosphate that we have seen in the previous diagram. Initiating reaction is the breakdown of the lipid phosphatidylinositol 4,5-phosphate or PIP2 producing inositol 1,4,5-triphosphate. This IP3 functions as a Ca2 plus signaling second messenger. All three phospholipase C derived signals regulate ion channels. Release of Ca2 plus from intracellular stores like endoplasmic reticulum, microsomal fractions, etc. is induced. Depletion of the endoplasmic reticulum Ca2 plus pool activates Ca2 plus influx from extracellular fluid through the plasma membrane. It shows what is explained here. The TRH receptors, TRH bind to the receptor, G protein activated, phospholipase C, the lipid metabolizing enzyme phospholipase C is activated, and this causes three types of messenger systems. One involving phosphatidylinositol 4,5-phosphate, then diacylglycerol, and inositol 1,4,5-triphosphate IP3. This activation of phospholipase C activate three messenger systems or three routes, and finally Ca2 plus is released. Ca2 plus from the storehouses like endoplasmic reticulum, microsomal fractions, etc. will be released. The release of Ca2 plus from the endoplasmic reticulum reduces the pool size. As a result, this will activate the movement of Ca2 plus ions from the extracellular fluid into the cytoplasm. And this activation, this release of Ca2 plus will activate protein kinase C, which results in downstream reactions, which results in the transcription of TSH. mRNA is produced. Using the mRNA as a template, thyroid stimulating hormone will be produced, which will be stored in vesicles. The vesicles move towards the plasma membrane, break open and release the TSH into the blood. Secretion of thyrotropin releasing hormone, and hence TSH is inhibited by high blood levels of thyroid hormones in a classical negative feedback loop. When thyroid hormones are present in enough quantity or in excess quantity, it will cause a negative feedback on the pituitary as well as on the hypothalamus. As a result, hypothalamus will produce less TRH, thyrotropin releasing hormone, and pituitary will produce less amount of TSH, thyroid stimulating hormone. A high concentration of TSH results in increased synthesis and release of T3 and T4 into the circulation. Low concentration of TSH lowers thyroid hormone synthesis and secretion. Effect of or actions of thyroid stimulating hormone. TSH is released into the blood. TSH binds to the thyroid stimulating hormone receptor on the basolateral aspect of the thyroid follicular cells. So this is a thyroid follicle. The thyroid follicular cell on the basolateral side. The receptors are present on the basolateral side, and this hormone binds to the receptors on the basolateral side. The TSH receptor is a G protein coupled receptor, and its activation leads to the activation of adenylyl cyclase and increase in the intracellular levels of cyclic AMP. The increased cyclic AMP activates protein kinase A. The activated protein kinase A phosphorylates different proteins to modify their functions. So TSH is released into the blood, which reaches the thyroid follicle, and it is attached to the receptors on the basal and lateral side of the thyroid follicular cells. So this is an enlarged view of the thyroid follicular cell. The TSH receptor is attached to the G protein. The attachment of TSH to the receptor stimulates adenylyl cyclase. This adenylyl cyclase helps in the conversion of ATP to cyclic AMP. This will activate protein kinase A. This activated protein kinase A phosphorylates proteins and make them active, which produces the desired physiological effect. Formation and release of T3 and T4. Monoiodotyrosine, that is a tyrosine molecule with one iodine atom, and diiodotyrosine, tyrosine molecule with two atoms of iodine are generated in thyrocytes. Monoiodotyrosine and diiodotyrosine are generated in thyrocytes. These molecules have iodine atoms in them. MIT has single iodine atom. DIT, diiodotyrosine, has two iodine atoms in one molecule. Thyroid peroxidase, TPO, combines iodinated tyrosine residues. The thyroid hormones are produced by the combination. Thyroid hormones are produced by combining MIT and DIT depending upon the requirement, and the enzyme involved is thyroid peroxidase. This enzyme combines iodinated tyrosine residues to make triiodothyronine and tetraiodothyronine. One each of MIT and DIT join to form T3. Monoiodotyrosine has one iodine atom and diiodothyronine has two iodine atoms. When these two are combined, the resultant product T3 has three iodine atoms. And two of the DIT molecules, diiodothyronine molecules, combine. So each diiodothyronine has two iodine atoms, and when two are combined,
the resultant molecule has 4 iodine atoms. Thyroid hormones are bound to thyroglobulin and stored in the follicular lumen. Release of thyroid hormone. Thyrocytes uptake iodinated thyroglobulin via endocytosis. Lysosome fuse with endosomes containing iodinated thyroglobulin. Thyroglobulin is cleaved into MIT, monoiodothyronine, diiodothyronine, and T3 and T4 by proteolytic enzymes in the endolysosomes. T3 is around 20% and T4 is about 80% are released into the fenestrated capillary networks via the transporter MCT8. So these two hormones are released into the capillary network. The capillary has got fenestrae. So the released molecules can easily enter into the capillaries. And the release is through a transporter named MCT8. DIT and MIT produced by the cleavage of thyroglobulin is now not needed. So the iodine will be removed from them by deiodinase enzymes. This removed iodine can be salvaged and redistributed to an intracellular iodine pool. So they can be reused. T3 and T4. Thyroxine. T4 is the predominant secretory product of the thyroid gland. Only 20% of the circulating T3 are secreted directly by the thyroid. In the periphery, T4 is deiodinated, supplying roughly 80% of the circulating triiodothyronine. Synthesis and release of T4 and T3 are inhibited when hormone levels in plasma exceeds a preset level. So sort of negative feedback mechanism. To a large extent, thyrotropin releasing hormone from the pituitary controls the release of thyroid hormone from the thyroid glands. TRH from the pituitary set the level of thyroid hormones in blood through pituitary thyroid axis. This contributes to the maintenance of homeostasis of the plasma hormone levels. To a large extent, the hormone level in blood is controlled by the thyrotropin releasing hormone from the hypothalamus. So this hormone controls the production of thyroid stimulating hormone by pituitary, and hence in turn it controls pituitary thyroid axis. The TRH control pituitary thyroid axis and set a point for the level of thyroid hormones in blood. This helps in the maintenance of homeostasis of this hormone level in the plasma. In the pituitary, TRH regulates not only the secretion of TSH, but also its bioactivity required for correct receptor binding. So that role is also there for the thyrotropin releasing hormone. It induces secretion of TSH and also its bioactivity, which is required for correct receptor binding. So TRH helps in the secretion of TSH as well as helps in the correct binding of TSH with the receptors. Sort of thyroid hormones. Thyroid hormones are lipophilic, lipid soluble, and circulate bound to the transport proteins. So lipid soluble, they are not soluble in the blood or cytoplasm. As a result, they are bound to proteins, and there is lipoprotein is transported across the body. Thyroid hormones are lipophilic. They are lipid soluble hormones. They are not water soluble, and hence its transport by blood is little bit difficult. As a result, it will be bound to protein molecule and lipoproteins are produced. These lipoproteins are transported in blood. Transporter proteins include thyroxine binding globulin, TBG, transthyretin, and albumin. Only a fraction, approximately less than 0.2% of the thyroid hormone is unbound and active. Normally, these hormones remain bound to proteins. When they need to be activated, they are released from the protein binding and they become active. On reaching the target site, T3 and T4 can dissociate from their binding protein and they become active. These hormones can enter cells by diffusion or carrier-mediated transport. Binding of these hormones to the receptors. Thyroid receptors are transcription factors that can bind to both T3 and T4. They have a much higher affinity to T3 than T4. As a result, T4 is relatively inactive. We have seen that 80% of the hormone produces T4 and T3 is more active because of its high affinity to the receptors. When T3 is needed, deiodinase enzymes can convert T4 to active T3 by removing one iodine atoms. Or it can also convert to inactive reverse T3. Physiological roles of thyroid hormones. Thyroid hormone affects virtually every organs and every type of cells in our body. In general, the thyroid hormone binds to its intranuclear receptors. This activates the genes for increasing metabolic rate and thermogenesis. Increasing metabolic rate involves increased oxygen and energy consumption. Thyroid hormone may induce lipolysis or lipid synthesis depending upon metabolism, body's requirement. When body need lipolysis, it will induce lipolysis. When body need to store more lipids, lipid synthesis will be induced. Thyroid hormones stimulate the metabolism of carbohydrates and anabolism or catabolism of proteins depending upon the situation. Thyroid hormone has a permissive effect on catecholamines. Only in presence of thyroid hormone, the catecholamines can cause its full effects on cells. That is permissive effect. In children, thyroid hormones act synergistically with growth hormone to stimulate bone growth. Effect of thyroid hormones on central nervous system. During the prenatal period, thyroid hormones are needed for the maturation of brain. So if thyroid hormone is less during the prenatal period, it will affect the brain development. In adults, thyroid hormones can affect mood. Hyperthyroidism can lead to hyperexcitability and irritability, and at the same time, hypothyroidism can cause impaired memory, slowed speech, and sleepiness. Thyroid hormone affects fertility, ovulation, and menstruation. Effect on different organs. Heart. Thyroid hormone have a permissive effect on catecholamines. Only in presence of thyroid hormone, catecholamines exerts its full effect. It increases the expression of beta receptors to increase heart rate, stroke volume, cardiac output, and contractility. Lungs. Thyroid hormones stimulate the respiratory centers and increases perfusion and oxygenation. Skeletal muscles. Increased development of type 2 muscle fibers. These muscle fibers are fast twitch muscle fibers capable of fast and powerful contractions. The role of TH on physiological processes. Metabolism. Thyroid hormone increases the basal metabolic rate, increases the gene expression of Na plus K plus ATPase in different tissues, leading to increased oxygen consumption, respiration rate, and body temperature. Thyroid hormone do not change the blood glucose level, but they can cause increased glucose reabsorption, gluconeogenesis, glycogen synthesis, and glucose oxidation. Growth during childhood is also influenced by thyroid hormone. TH acts synergistically with growth hormone to stimulate bone growth. Bone growth means more growth of the individual. It induces chondrocytes, osteoblasts, and osteoclasts. As a result, bone growth will be induced. Also helps in the brain maturation by axonal growth and the formation of myelin. It also helps, TH also helps with brain maturation by axonal growth and the formation of the myelin sheath. How is TH exerting its action? Receptors for T3 remain bound to the DNA in nucleus. T3 or T4 then bind to the nuclear alpha or beta receptors in the tissue. Cause activation of transcription factors. Activation of transcription factors activates certain genes and cell-specific responses are produced. After their use, TH will be taken to the liver and degraded by sulfation and glucuronidation, and the degraded products of TH will be excreted in the bile. Degraded products of TH will be excreted in the bile. Thank you. You need to answer the following question. Hypothalamus acts as a link between nervous system and endocrine system. Explain. Hypothalamus controls anterior pituitary secretions. How is that control effected? How is water reabsorption controlled by hormones? Explain process of stimulation or inhibition of T3 and T4 secretion. What are tropic hormones? Thank you.